So learning outcome for today's lecture is for you to be able to describe the general functions of proteins and to identify and classify the 20 amino acids present in proteins, to discuss the ionization of amino acids, and also to describe various techniques for protein purification. So I have divided different types of subtopic for today's lecture. So we learn about the introduction to proteins, general functions of proteins, amino acid structure and properties, amino acid classifications, ionization and acid-base properties, the peptides and the proteins, and also the techniques for protein purification. So we will cover all these 7 subtopics today. Okay, so the first one, what is protein? So last week we learned about the inorganic macromolecules, right? So there are different types of macromolecules: carbohydrates, protein, nucleic acid, lipids, right? Okay, so proteins it's a type of inorganic biological macromolecule. Macro, it is because it is big. It's large, complex molecule for normal functioning of cells. And its building block or basic unit is actually the amino acid. And amino acid form peptide bonds of long chains of proteins. And there are different functions of proteins, for example, as a catalyst, storage and transport, structural. It involved in mechanical work, decoding informations, and also as a hormones or hormones receptor. So let's see how protein works in our body. So in immune system, it is secreted as antibodies to fight the pathogens, and also released as signaling proteins, for example, cytokines, stimulated by the inflammatory response, so that for the communication with other cells. And also proteins in blood as a hemoglobin, fibrinogens, albumin, and also as an enzyme, different types of enzyme that involve in, for example, digestive enzyme helps in breaking down foods. And also structure of cell membrane, the channel for substances to move through the membrane, and also it can be the enzyme receptors and other type of proteins that involve in cell membrane. And for structural protein, for example, the cytoskeleton, keratin, collagen, elastin, and protein in muscle, for example, actin, myosin, myoglobin, ferritin. Okay, that is different types of protein in our body. And the basic unit of protein is made up by the amino acid. So what is amino acid? It composed of amino group here, and also the carboxylic acid, the carboxyl group, attached to what we call it as a alpha carbon. So this is how alpha carbon bonded with 4 different types of molecules, which is the amino, hydrogen, carboxyl group, and also the R. R is actually a side chain of the group. It could be any group that determine the functions of the specific amino acid. So you have to know that amino acid is coexist in 2 forms, which is in ionic, the zwitterion form, or also in unionized form. So you need to know as well the configurations of the amino acid. So I mentioned just now alpha carbon, here the alpha carbon attached to 4 different groups, right? The hydrogen, the COOH, the carboxyl group, amino group, and also the R group. So the attachment of different groups, we call it the chiral, or it produced in a form of stereoisomer of the amino acid. And the 2 isomer of mirror image exist in a form of L and D. You see the different image of one amino acid can be in a form of D or L. It's a mirror image of each other. So we call it as an enantiomer. And you have to know that all amino acids found in proteins are in L configuration except for the glycine. So the glycine is the simplest amino acid. You can see here, the C carbon, the alpha carbon here, does not attach to only two, like amino and carboxyl, but it attached to two hydrogen. It has to be four different groups in order to be a stereoisomer. So if there are a common group together, that it cannot form a stereoisomer. It is not chiral or stereoisomeric. So except for glycine, all of the alpha amino acid form are found in protein exist in L configuration. Okay, so far clear everyone? I think this is a basic of amino acid structure. I think you all should know already. Okay, so now move to amino acid classification. So how amino acid is being classified? So I divided into three different division here. For example, classification based on the R side chain structure. So it can be non-polar, polar, acidic, or basic based on the R, its R structure that attached to the alpha carbon. And also other R side chain structural classification. It can be aromatic, aliphatic, and also sulfur containing group. And other different classification which is not related to the structural of our side chain is that it can be classified based on nutritional requirement, whether it is essential or non-essential amino acid. So let's look at first how it is classified based on the nutritional requirement. So you have to know that there is two types of nutritional requirement, essential and essential of amino acid. And essential amino acid is actually a group of amino acid which is not synthesized in our body, that we have to obtain it from our diet. So this is the type of essential amino acid. And non-essential amino acid is a group of amino acid that already synthesized in our body. Okay, so this is a type of non-essential amino acid. Okay, most importantly, you have to know the classification based on the structural properties of the R side chain of the amino acid. So it can be about 20 amino acid. It can be divided whether it is hydrophobic, non-polar, or hydrophilic, the polar. So for non-polar, you can further divide it whether it has the alkyl group or aliphatic. There are 7 of them in this group, and sulfur containing group is for cysteine, and aromatic, which has the aromatic group in the R side chain, phenylalanine, tryptophan, and also tyrosine. And also for hydrophilic, the polar amino acid can be divided into acidic or basic, and also the neutral or uncharged amino acid. Okay, let's see one by one. Okay, first, this is the structure of 20 common amino acid. So you can see different types of like tryptophan here. So tryptophan, it has the aromatic group. So aromatic ring present for in the R side chain that attached to the chiral carbon, alpha carbon of the amino acid. So this is a tryptophan and phenylalanine, and another one is tyrosine. So 3 of them has the aromatic ring structure for their R side chain. Okay, so you can go back and study different types of amino acid structure, and I think you can easily can understand each classification based on the R structure. Okay. Okay, so for non-polar aliphatic amino acid, there are 7 of it, right? The glycine, alanine, proline, valine, methionine, isoleucine, and leucine. So what is it? It has a non-polar, not a non-polar, but lack of polarity functional group in R side chain. So that is why it becomes the hydrophobic. So it usually form a cluster together within the interior of the proteins that it made, and it stabilize the protein structure via the hydrophobic interaction. Phenylalanine, tyrosine, and tryptophan, all of them contain the aromatic group. Okay, and the side chain here are very hydrophobic. So that is why it is belong in the hydrophobic group, the non-polar group. And also sulfur containing amino acid, which have the sulfhydryl group at the outside chain. It is a weak polarity and also uncharged at neutral pH. And 2-cysteine can form a disulfide bond. Okay, this is a disulfide bond. All right. And number 4, this is number 3, number 4, polar uncharged amino acid. Neutral. So the R structure for asparagine, glutamine, threonine, serine has the polar uncharged structure, and it can form a hydrogen bond along with water. And serine and threonine actually have the hydroxyl group for their outside chain, and asparagine and glutamine has the amide group where the nitrogen bind to the carbon, forming the amide group here at the side chain. So as you can see here, there is an amine group at the outside chain. Right, everyone understand? All right, number 5, basic polar positively charged amino acid. Okay, so the R chain should be hydrophilic because it's polar, right? But it's basic. Okay, so the R group of lysine and arginine are fully positively charged at neutral pH. And histidine, the R group, contain the aromatic imidazole group that is partially positive charge at neutral pH. So that is the difference. This is as a linear structural, and this is as the aromatic ring in histidine. Number 6 is acidic polar negatively charged amino acid. So the R group of both aspartate and glutamate contain the carboxyl group. Here, carboxyl group. This carboxyl group is for the backbone of the amino structure that attached to the carbon, but it also has the carboxyl group that attached as its side chain. All right, so that is for aspartate and glutamate. Okay, so I finished with the classification. So I hopefully you all can follow and can differentiate different types of classification of amino acid. So are you following? Is everyone okay? It's
Yes,
3
doctor.
p.m.
Yes, Doctor.
So another property of amino acid you should know is about the ionization. So everyone familiar with zwitterion?
No,
Yes.
doctor. No.
Yes, okay. No? It's okay. So for zwitterion, so we should know the 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 characteristic of amino acid that it can exist both in zwitterion, the ionized form, or in unionized form. So if you can see here, the structure of amino acid, that both the amino and carboxylic can exist as a basic group or acidic group. So it can be ionized when come interact with water. So the amino group belongs to the basic group. It can be basic, and the carboxyl group can be in a acidic group. So you should know that the pH at which amino acid exists as deuterion is where there is no net charge of them. Here, positive and negative balance. Okay, so that pH we call it as isoelectric pH or isoelectric point. The pH where both basic group and acidic group, or carboxyl and amino group, have no net charge of the pH. So balance, positive and negative is balance. You will see how the unbalance of the positively charged is affected by the pH later. Okay, so deuterion can be either basic or acidic, depends on the pH. Okay, so let's see, let's look at the picture for the acid-base property of amino acid. So at pH 7, the net charge of the amino acid is 0. Because the amino group is positively charged, carboxyl group is negatively charged. So it is in zwitterion phase or state, which we call it as a neutral, no net charge for this amino acid. Okay, so when simple amino acid, for example, alanine, is dissolved in water at neutral pH, so its carboxyl group is negatively charged and amino group is positively charged. So the net charge is 0. It is what we call it in a zwitterion state. So that is the zwitterion. So what happened when the molecule become or is in acidic medium? So this COO, the carboxyl group will pick up. So in low pH condition, there are abundant of H or hydrogen ion. The carboxyl group will pick up the hydrogen ion. So it will become uncharged. So when carboxyl group uncharged, so there is positively charged of amino. So the charge is unbalanced. So the net charge is actually +1. So it is called the positively charged amino acid in a cationic form. Do you understand or not? And then when in basic medium or when there is high pH, the amino group there is a will remove here. So when there is it exposed to basic medium or high pH, the amino group here will remove the hydrogen ion, so it will become neutral, and that the carboxyl will leave as a negatively charged. So the whole amino acid is negatively charged and it is in an ionic form, so that in electrophoresis it is more attracted to anode. And this is during electrophoresis for protein separation, it is more attracted to cathode. Maybe you still do not understand, but when we learn about the electrophoresis later, maybe you can catch up. But I hope you can understand different types, different form of amino acid in different pH level. So in low pH, pH 7, the zwitterion phases or stage, and also when there is increase of the pH in a basic medium. So okay everyone, so let's move on to the peptide and protein. So what is peptide? Peptide is a short polymer of amino acid joined by peptide bond. So two amino acid bind together, we call it as a dipeptide, and 3 amino acid tripeptide, 12 to 20 amino acid, we call it oligopeptide, and more than 50 amino acid bind together to form by the peptide bond, forming the polypeptide. Okay, for example, amino acid lysine here, and it will bind to different types of amino acid to become the angiotensin, the peptide. And the peptide, accumulation of peptide will form the immunoglobulin, the IgG. And also protein can be degraded. It can conjugate it to become a larger molecule, and also it can be degraded to become a smaller molecule, like from protein degraded into the peptide and then degraded again to become the basic unit, its basic unit. Okay. Okay, what is the peptide bond? So amino acid are linked together by the amide group called a peptide bond. And you have to know that during the process of protein synthesis, the carboxyl of amino group at the end of the growing polypeptide, the active site, we react with the amino group of incoming amino acid, and by releasing the water molecule, it forms the peptide bond. For example, this is one amino acid and another amino acid. The carboxyl group from one amino acid will interact with the amino group of another amino acid and releasing water molecule, that it will form the peptide bond. Okay. So this is an example how the alanine bind to the glycine. Okay, so this is a carboxyl group from the alanine and amino group from the lysine. And from the process of dehydration, removing of water molecule, so it is bound with peptide bond here. So let link the alanine and glycine to become alanyl glycine. So it is a dipeptide. Two different or two amino acid bind together. So everyone okay with peptide bond?
Okay,
Right.
doctor.
All right.
Okay, doctor.
So this is very basic. Okay, the last topic for today, I mean the subtopic, is for you to know. So you already know the protein itself. It's a large complex molecules and it is made up by its basic unit, the amino acid, right? And you should know there is a technique. There are actually several hundreds of proteins existed. And you should know that there is a mixture of protein and how we can separate and purify those protein into smaller group into according to its size or according to its charge. So there is a technique. It's analytical technique. So what is protein purification? We call it as a protein purification. It's a series of processes to isolate a single or multiple type of proteins from a complex mixture of proteins. So the protein can be isolated based on their property like solubility, molecular size, molecular charge, polarity, and also specificity. Okay. This is the types. I give an example here how to isolate a protein based on its solubility. So we can use salt precipitation, adsorption chromatography, and also we can separate the protein according to its molecular size. So we use the dialysis and ultrafiltration and also size exclusion chromatography. We can use gel electrophoresis or ultracentrifugation. And also we can separate protein by its molecular charge. So that one known as ion charge chromatography or electrophoresis. And also if we want to separate the protein based on its polarity, we can use a paper chromatography. And also specific, like there is a ligand binding, we can use the affinity chromatography and immunopurification. So this is two types of more advanced technique that is used to separate protein mixture. Okay, let's go one by one. So what is the dialysis? So let's say you have a mixture of proteins like in your extract. So you want to separate proteins of your interest. So you separate it based on the size. So by using a semi-permeable membrane here, which have pores that less than macro molecular dimension, less than your protein of interest, so that your protein interest will be contained or accumulated in the membrane. So unwanted protein, the smaller size protein will be go out. And then you will take the semi-permeable membrane out, and then your protein of interest is trapped in this semi-permeable membrane. So this is one type of how you can isolate the proteins. So this is the blue one, the larger macromolecule is your protein interest. So it will be trapped here. So this is one type. So another type is ion exchange chromatography, where we isolate the protein based on its charges. Okay, let's say this is a column and there is a resin here, the substance that the resin, it depends on your protein or interests actually. So the resin will bound to negative functional group, and then the positive charged protein that is not needed in the mixture will be washed out. Okay, so the protein will move through the column at rate determined by their net charges. With cation exchanger, protein with more negative net charge move faster and elute earlier. So cation will bind to the negative functional group and it will be elute. So let's say your protein is positively charged, so you should use the resin anion. So we will put the resin anion bound to the positively charged proteins, and then will be elute. So that is how we separate the protein based on its charges. So this is actually a very advanced technique. Just want to show you the principles of how protein can be separated based on their properties. So this one, specific protein that needed. So this is a chromatography technique. So this is a column. So there's a resin in the column contain a ligand that is bound to your protein interest. Let's say I want to separate the protein X. So I have a specific ligand that bind to the protein X. Okay. So the unwanted protein that is not bound to my ligand will be washed through the column, so that later I will only collect the protein that is bind to the ligand, which is my protein of interest. Right. And the simplest one is a PAGE, SDSPAGE, polyacrylamide gel electrophoresis, sodium doxydyl sulfate, polyacrylamide gel electrophoresis. So what it do is that it separates the protein using electric charge, so that the main principle of SDS space is actually to estimate the molecular weight of your protein sample. So initially, protein will be dissolved in a buffer containing SDS, and it will be separated on a polyacrylamide gel by electrophoresis. And the locations of protein on the gel is determined by staining with Commercy blue. So you will see the blue staining here. And then we can expect or estimate the protein molecular weight by comparing the band that appear here for each protein with marker. So marker, is a standard. We already know the molecular weight of each band in the marker. So let's say this is 45,000 Dalton, and we can expect if our band lies in between here, so we expect it is like 45, 46, 45, 45,000 Dalton, okay, according to the standard. So this is PAGE is usually used to estimate the molecular weight of the protein sample. Okay, this is one type of protein. So if you can see the different layer of the proteins in PAGE here is actually different proteins. So this is different protein, different proteins, different types of proteins. So protein accumulated here could be the same type of protein. So it is abundant in the sample. Okay. All right. So summary for today's lecture is that you have to know both alpha amino acid and non-alpha amino acid occur in nature, but only proteins synthesized using alpha amino acid except for the glycine. Okay, I already explained it in the amino acid configuration just now, right? And also you should know that the art group, the side chain of the amino acid determine its property and also the classification of the amino acid. So it can be classified into basic or acidic, polar, or aromatic, aliphatic, and sulfur containing, which is a hydrophobic group. And also protein mixture can be isolated and purified based on their properties such as solubility, molecular size, molecular charge, polarity, and also its specificity. So as for this number 3, as I said, as long as you know different types of, when I ask what is the protein purification technique that you learn to isolate the mixture of proteins, and you can answer like to separate the protein based on its molecular size. So we use which technique? So the affinity chromatography is actually what? Based on specificity, right? All right. So that's all for today. It's a very basic lecture for amino acid and peptide.